You were once one cell big. How did you come to be several thousand trillion cells big? In a nutshell, the answer is by cell division. And I think that whoever sent this set of text messages, though their spelling is a little wanting, did a pretty good job of summarizing it. Basically, you start with one cell which grows in size and then pinches in half until it forms two smaller daughter cells that are identical to the parent cell. There are many regions that biologists, and indeed us, should care about cell division. One reason is it explains how we came to be many cells from one. But also, when cell division is not regulated properly, some unusual things can happen in our bodies. Tumors form when the cell cycle is not very well regulated, and so cells start to divide and divide out of control, as happened on this gentleman's face right here in his salivary gland. Now, some tumors, though they don't look great, are not that threatening to our livelihoods. They're called benign tumors. Benign means harmless, and in this case, this patient was able to survive for another 30 years after having this benign tumor removed. It did not have any adverse effect on his health. Because the tumor in his salivary gland was so contained, it was surgically removed without any remnants and did not affect his health. Now, that's just benign tumors. Sometimes tumors can form and then spread, and that's when things become very problematic. Let's take, for example, a common spreading kind of tumor that occurs inside your large intestine or colon. Sometimes when a tumor forms on the inside of your large intestine, like right here, you can see a bunch of cells that are dividing way too quickly. If they continue to divide, they might break through the surface of the large intestine and start moving inward until eventually they reach a blood vessel. And that's really bad news, because then the cancer cells can jump into the blood vessel, swim through the blood vessel until they get to a new tissue, some new organ elsewhere, and then start to crawl out of those blood vessels into the new tissue and form a new tumor in a new area. This process is called metastasis. And when metastasis occurs, this abnormal kind of growth gets a new name. It's called cancer. In the case of colon cancer, let's say that a tumor forms on the inside of the colon, as is shown right here. If that tumor is able to grow large enough that cancer cells can hitch a ride into this hepatic portal vessel right here, they'll take that upwards here until they get to this large organ at the top, the liver. The liver helps us remove toxins from our blood as well as metabolize our food and generate bile to neutralize stomach acid. It has so many important functions. Needless to say, if a bunch of metastases or small tumors start to form in this liver thanks to the traveling cancer from the large intestine, then our liver will be taken over by cancer metastases until there's no normal liver left and we could die from liver failure. So when the cell cycle is not properly regulated, cell division can occur too fast, leading to the formation of a tumor, which can lead to cancer if metastasis occurs. Yikes. Okay, so then why do cells divide at all? Why not just exist like this amoeba, growing by engulfing a small little food item right here using some pseudopods, and then slowly let your cells get larger and larger until you turn into a huge amorphous blob and just exist that way. Then you have no cell division and no cancer, right? Sounds great. Well, there's a problem with that. As cells get larger, they get less efficient. This is illustrated in this diagram pretty well. If you imagine one smaller cell right here, and then another cell that's twice as big, and then another cell that's 3 times as big as the first one, you can see why large cells are much less efficient than small cells. Take, for example, the role of the nucleus, which contains, as you know, DNA. In a small cell, that DNA can send out just enough messages to boss around the entire cell. But as you get to larger and larger cells, this poor nucleus right here is going to be pretty overwhelmed. It's not going to be able to send out enough messages to... And this nucleus, holy smokes, it's going to be really overwhelmed. It has to send so many messages out into the huge volume of this cell in order to boss the entire thing around. So this cell over here on the right is not going to be very efficient for that reason. When it comes to absorbing nutrients, also the cell on the left is going to be a lot more efficient than the cell on the right, because the large cell has a very much larger volume that it needs to feed in comparison to the surface area, that is the outside of the cell, that is available to feed it. What do I mean by that? Well, the small cell has a surface area of just 1 by 1 times 6, because there are 6 sides on a cube, so that gives you a surface area of 6 centimeters cubed. The next cell would be 2 times 2 times 6, giving 24 centimeters, and the last one 3 times 3 times 6, giving 54 centimeters. Okay, so you look at this cell and you say, the surface area is increasing as the cell gets bigger. That makes sense. And why would that be a problem? At least this cell has a lot of surface area, 54 centimeters squared, in order to take in food. The problem is the volume. So this tiny cell right here has a tiny volume, just 1 centimeter cubed, 1 centimeter by 1 centimeter by 1 centimeter. So it only has 1 centimeter cubed worth of space in it that needs to be filled with food. On the other hand, this cell is 2 times 2 times 2 in volume, so that's 8 centimeters cubed. That's quite a bit larger, so a lot more food, 8 times more food. And then if you take the next one, we go 3 centimeters times 3 centimeters times 3 centimeters, that's 27 centimeters cubed. And if you know your cubes, you know the next one is 64, and the next one is 125. The point is, the volume increases much faster than the surface area does as the cells get bigger. In other words, as cells get bigger, their demands for food grow much faster than their ability to obtain that food. So don't worry, the world is not going to end with one huge cell, the blob, coming and trying to engulf everything, because there's no way that that blob could take in enough food by its surface area to feed its vast volume. And to avoid this inefficiency, cells divide. Cell division can be simple in some cases and harder in others. It's particularly easy if you are a prokaryote, as you guys remember. Pro means before, karyo means nucleus, so prokaryotes don't have a nucleus. Instead, they just have a single wound-up circular chromosome of DNA depicted right here that can then be copied, moved to opposite sides of the cell, and then the cell wall and cell membrane can grow down and in until eventually it divides into 2 cells, and voila, prokaryotic cell division is completed. It is called, therefore, binary fission, binary meaning 2 for the 2 cells, and fission meaning splitting in half. And one thing that's very distinct about binary fission is that the 2 daughter cells are completely genetically identical to the mother cell that started off up here. Though eukaryotes also have a phase of growth, a phase of copying their DNA, and then a phase of dividing, the eukaryotic cell cycle is indeed much more complicated. It starts off with this first cell growth phase known as G1, then moves into the DNA replication stage. To replicate means to copy. This stage is also known as S phase, and the S stands for synthesis, or making, of DNA. So it copies its DNA, and then it goes into its final phase of growth, G2, in which the cell grows even larger, and starts to move organelles to either side of the cell, preparing for division. And then we've got the division itself. Often this part of the cell cycle is simply called cell division. Sometimes it's also called M phase, because during M phase, the cell needs to go through the bother of dividing its nucleus. After all, this is a eukaryotic cell, which means that it does have a nucleus. And so division isn't just a simple chop in half here. It's, alright, take that nucleus, break it down, move copies of the DNA to either side, which happens in these phases here, prophase, metaphase, anaphase, telophase, which we will study more in later lectures. And then the actual cell gets broken apart in a phase called cytokinesis. So overall, the eukaryotic cell cycle, much more complex. We've got G1 for cell growth, S phase for copying that DNA, G2 for more growth and preparing for division, and then division itself, which has many subparts. All these other phases, the G1, the S phase, and the G2 together are referred to as interphase. So that's inter means between, so interphase is the phase between cell divisions where most cells spend their time.